endocrine system. So, as we said at the end of the last set of slides, hormones act in one of two ways, and that depends on the chemical nature, and the chemical nature of the hormone will help determine where the receptor is located, either on the surface of a cell or internally. So the water-soluble hormones, the amino acid-based ones, except for the thyroid hormone, have plasma membrane receptors. They work through a G protein second messenger system. They never make it into the cell. Whereas the lipid hormones, the steroid hormones and the thyroid hormones, diffuse across the cell membrane, contact their receptor within the cytoplasm, and go into the nucleus and work on genes within the nucleus. So these ones do enter the cell and actually have to enter the cell to do their job. So the amino acid-based hormones, except for the thyroid hormone, work through second messenger systems. And even if you don't remember the details of each of these second messenger systems down the road, when you hear second messenger system, you should think amplification. One receptor will turn on many responses inside the cell. So the two main second messenger systems are the cyclic AMP and the PIP2 calcium-based second messenger system. The cyclic AMP signaling mechanism, the first messenger, which is why we call it a second messenger system, is the hormone itself. The hormone binds to a receptor on the surface of a plasma membrane. That receptor activates a G protein, and that G protein turns on and off enzymes. And one of the important ones is adenylate cyclase. Adenylate cyclase's job is to take ATP and to turn it into AMP, so it removes two phosphates, and at the same time bends it into a circular structure. So we call that cyclic AMP. And that cyclic AMP is the second messenger. So one hormone is going to make big changes in the levels of cyclic AMP. Cyclic AMP works to activate kinases, and those kinases add a phosphate to other proteins, turning them on or turning them off. Those phosphorylated proteins, activated or inactivated, are going to cause that cell to do what it needs to do. To get rid of cyclic AMP, it's degraded by an enzyme called phosphodiesterase that gets rid of the cyclic AMP, stops the cascade, stops that amplification, and that's really the secret to second messenger systems. One hormone is going to activate a lot of cyclic AMP in this case. So we have a hormone binding to a receptor. That receptor is joined to a G protein. The G protein normally has GDP on it. It's an inactive state. GDP will be replaced by GTP, activating the protein. That protein will then hit another membrane bound protein called turning on cyclic AMP. One step at a time. So hormone, the first messenger, binds to the receptor. Second, the receptor activates a G protein, which means the GDP is removed and the GTP is added. And that, just like ATP for us, except it's a G, a guanine instead of an adenine. That G protein is going to activate this other membrane protein called adenylate cyclase, and when GTP bound G protein is stuck to adenylate cyclase turns on and ATP is converted to cyclic AMP. Cyclic AMP, the second messenger, then works to turn on protein kinases, and that protein kinase will hit other targets, turning on and turning off things that need to be done in response to this hormone. So again, the key here is one hormone, one first messenger, lots of second messenger, amplification. The other mechanism, the PIP calcium signaling mechanism, is going to be very similar. It's a second messenger system. The second messengers are different though, but it's still amplification, and that's the key thing I want you to remember about second messenger systems no matter where you see them. So the hormone activated G protein activates an enzyme called phospholipase C instead of adenylate cyclase, and that activated phospholipase C splits a membrane protein into called PIP2 into two second messengers. Those two second messengers are diacylglycerol and inositol triphosphate. Diacylglycerol does what cyclic AMP does. It works on protein kinases. Inositol triphosphate, IP3, causes calcium to be released from intracellular storage sites, and calcium will do things in the cell. The calcium is another second messenger. Calcium alters the enzyme activity and channels, and it binds to a regulatory protein called calmodulin. And when calcium is bound to calmodulin, it activates enzymes that amplify cellular responses. There are other signaling mechanisms out there. Cyclic GMP, like cyclic AMP, is a second messenger for some hormones. Other hormones work without second messenger systems. Insulin, for example, binds to a tyrosine kinase enzyme. And a tyrosine kinase puts phosphates onto tyrosine residues. Tyrosine's an amino acid in proteins. And that activated tyrosine kinase is a docking site for relay proteins that trigger responses. So not quite the same. The other type of hormones are those that get into the cell and turn on genes directly. So those lipid-soluble steroid hormones and thyroid hormones diffuse into the target cells and bind with intracellular receptors. The receptor hormone complex would then enter the nucleus, bind to a region of the DNA, and when you bind those proteins to the DNA, that causes that piece of DNA to become transcribed or copied into messenger RNA, and messenger RNA is the instructions to build specific proteins. So that messenger RNA will leave the nucleus, go to the cytoplasm, find a ribosome, and be built into its protein, whether it's an enzyme, a structural protein, or whatever kind of protein. It's going to work on metabolic activity, structural purposes, exported from the cell even. So in this case, a steroid hormone here is going to make its way through the plasma membrane and bind to a receptor inside. That receptor hormone combination gets into the nucleus. Into the nucleus, it finds specific binding sites on lengths of DNA, finds genes that it's going to turn on, and those genes are then turned on, which means they are translated or transcribed into messenger RNA, and those messenger RNAs are then exported from the nucleus, where they find ribosomes, and ribosomes will build these messenger RNAs into new proteins. And that brings us to how the levels of hormones are regulated. I'm going to save this for the next set of slides.